Okay, so let's talk about the biological aspects. Here are my disclosures. So many of you have heard about, and we've talked about repeatedly over the last couple of days, about precision medicine. And precision medicine, and I think Dr. Petralia put this quite well, he said it's the right treatment for the right patient at the right time, and this is what your clinicians will tell you. But in fact, what they often forget is for the right duration. And so for patients to proceed down this precision oncology route, pathway, there need to be some next generation imaging tools. And most of these are liquid tools, blood tests and like, biopsies, but there's also imaging. These next generation tools, molecular pathology and advanced imaging, allow you to pick the right treatment depending on the molecular pathology that is found using those tools and the distribution of the disease. And those also serve as biomarkers of tumor activity. So when you repeat these imaging tests in time, and then hopefully you can see if patients are not responding to treatment. This is called primary resistance, whether they're stable and then they progress, or whether they respond and then they progress. And these imaging tools will often give you an idea of when patients are progressing at an earlier time point than conventional imaging. And so if you repeat these tools again and again and again, you can get more treatment into the patient during their course and hopefully keep them better over a long period of time. And this is the paradigm of precision oncology as it is practiced today. So precision oncology requires us to have an idea about changing tumor biology over time. And sometimes blood tests don't tell you everything because it can't really depict heterogeneity of response. And that's where functional imaging comes in. So because functional imaging tells you in a quantitative way what is happening in the biology at each voxel in the body over time. So of course that's a challenge. And the multiparametricity, the ability to do multiparametric imaging tells you about habitats and how tumor habitats change over time. So if you look at a patient like this, so this is a patient with about 1.2 liters of metastatic prostate cancer in the castrate-naive setting. When you see this much cancer, not every site is the same, correct? So now we know that there are certain hallmarks of this tumor and this tumor distribution. And we know that these hallmarks are targetable by drugs. So we already have EGFR inhibitors, for example. We have anti-immune treatments. We have PARP inhibitors, et cetera, in the clinic. So these are targeting certain hallmarks. And so today we're going to talk about a few of these hallmarks. We're going to talk about proliferation, cellularity, metastasis, and death, which you can package into the same packet. We will also talk about looking at blood flow and about metabolism. Okay, so we know that for tumors to grow, hallmarks have to accumulate, as we've just said. The way these hallmarks accumulate is by mutations in regulatory genes. And the way these genes dysregulate changes across time. And so cancers are characterized by the accumulation of non-lethal mutations that allow the cells to grow in a crazy way. And these genes, you have to remember, are packaged into pathways. So, for example, in prostate cancer, the important pathways are things like the PI3 kinase pathway, the Wnt pathway, the AR pathway, et cetera, et cetera. So genes are bunched into pathways, and these pathways then drive the proliferation. And as the tumor advances, what you notice is that the number of genetic aberrations increases. So if you go from left to right, you'll see on the left is local regional disease. On the right, you have castrate-resistant disease. And I've got some of the pathways on the left-hand side. I don't expect you to be able to see this, but I want you to look at the number of dots. And you can see how, as you go from left, the tumor advances, so the number of genetic mutations increases. Okay? And so what you have to remember is not only as the tumor grows do you get increasing numbers of mutations, but that is also regulated by all the treatments that we give. So as we give treatments, tumors shrink and then tumors bounce back. And as they adapt to what you are giving, this is clearly a Darwinian process, what happens is you begin to see accumulation of mutations that are different from the very beginning. In other words, you get a kind of branching evolutionary tree that develops that you can actually see as you image. Okay, so the bottom line is, as tumors get bigger, you get multi-clonal proliferation. So the question is, can you see proliferation in life? Can you see
So what we ended up building was basically a petri dish,
okay?
except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, porous and thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads, until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
Okay, so I've given you some idea about how you might be able to see bacteria growing in a petri dish. How's that useful to you, right? Well, can you see this happening in humans? Well, let's imagine that the human body is the petri dish. Let's imagine that the bacteria are tumor cells. Let's imagine that the antibiotic is the anti-cancer therapy. And what do you have in your pocket? You have the light camera, which is, of course, your medical imaging tool. Now, what imaging tool would you use to look at proliferation? Well, if you go down this list, you can see to look at cell density and cell proliferation, you can use diffusion imaging. Diffusion imaging, you know, looks at cellularity of tissues. And it also looks at tissue viability in response to therapy. All of us have scanners that enables us to look at cellularity across the entire body. And this is the same patient that I showed you previously with the rotating color image. So you can look at heterogeneity in response also. So let's just have a look at this woman who has metastatic breast cancer and is being treated with letrozole. So that's the antibiotic. And the petri dish is, of course, this particular patient, and our technology is whole body MRI. So you can see at the beginning, we have, no, you can't see that. Let me see if there we go. So in the beginning, you can see she has a breast lesion in her left breast, 2 small metastases in the bone. You can see that after some treatment, things seem to get better. But in fact, looking very carefully, you'll notice that there is a little residual abnormality in the left breast. So this is an area of resistance. What I'm going to do is I'm going to just blow that up for you so you can look at this again. Okay, so you can see there's an area of resistance in the breast, just there it is. And look what happens over time. So over time, on the next scan, what we see, axillary lymph nodes are beginning to grow. And then the next time, the treatment never changes, remember? So the axillary lymph node begins to grow, the bone disease reappears, there are new liver metastases on the third time point. On the fourth time point, we have completely lost control. We've completely lost control. And you can see that she develops more and more metastatic disease. Now, why did the patient carry on the same treatment? That's the interesting question, right? This is clearly clonal evolution. This tumor has resisted what we gave it. We saw the evolution come through, but we never changed our treatment because she didn't want any chemotherapy. So she had ER-positive breast cancer, and at that stage in 2017, we didn't have palbociclib, et cetera, that we could give her, and she refused any other treatment. So it gave us this opportunity to look at tumor evolution within the body. Now, we know that when you do diffusion imaging and you drill bones and you look at the number of cells, the ADC value correlates directly with the number of cells down the trophine biopsy. So we know it's important, and it tells us something that's quite interesting. Diffusion imaging also allows you to look at cell death, and we're interested in cell death. So a shift in the histogram, a reduction in signal intensity of a high B-value image tells you about cellularity. Not only that, but you can also begin to map where cells are dying and where cells are alive. So in this particular depiction, you can see that red means alive, green means it's dead, and the middle is yellow, it's dying. So alive, dying, dead. So we've deliberately devised a traffic light system for our clinicians to tell them what's alive, what's dying, what's dead. And if you look very carefully now, you can see that this is a heterogeneous response. Some areas have responded very well, other areas have not responded very well at all. And now we can begin to drill those red areas and see what mutations are occurring in those red areas. So in fact, this has been done. So in this particular patient, this is a publication in Cancer Discovery, I think it was last year. What they did was they drilled the bones, and you can see in the first examination, they drilled the left iliac bone and found a BRCA mutation. Now, we know BRCA mutations are actionable. We have PARP inhibitors in the clinic, so they treated with PARP inhibitors, and you can see at the second time point, the ADC value goes up. That's a therapeutic response. On the third occasion, you can see it's the other iliac bone that is now cellular. And when that was drilled, the BRCA mutation had reversed. So cancer cells will try to adapt themselves to the treatments that we give. And we can guide biopsies to be able to see what other actionable mutations are present. And if you do imaging like this, and you can see on this particular occasion, I've done this 7 times in this patient with metastatic prostate cancer, just using that very simple classification, alive, dying, and dead, you can begin to follow this patient across time, and you can quantify the amount of tumor that is alive, and that's the red value. So you can see the patient starts off at 95%, and then after 4 cycles of chemotherapy, it goes to 24%, and then the next one, it's 28%. So actually giving 5 more cycles made no difference for this particular patient. And as you might imagine, this patient's going to bounce back, and everything becomes red again. It goes back up to 82%. We change treatment to cabazitaxel, no response at all. It goes up, and then we change to abiraterone on the last occasion, and you can see it's come down. So you can use these metrics and begin to titrate treatment according to the response, just in this particular instance by looking at the number of alive cells. Now, if you want to see other illustrations of other patients where we've done this, and just watch a couple of videos on my YouTube channel. There's the plug for my video channel. You can see, I've done a number of patients where I've shown 15, 16, 17 examinations and how you can follow this over time. The other interesting thing is, as tumors evolve and they become clonally diverse, we know that other things also change, not just proliferation. So we know that there are changes in metabolism and in blood flow, for example. And we know that metabolic changes and blood flow changes occurs before the proliferation change. So for example, we can look at blood flow by using something as simple as a CT scan. And we talked about the Choi criteria a couple of days ago, how a decrease in the enhancement is actually a change in perfusion, and how that can relate to a blood flow change. Now, the interesting thing is, why does that happen? That happens because as you give anti-angiogenic therapies, you get vascular pruning. As you get vascular pruning, the enhancement goes down. The enhancement goes down, but if you look at the edges of these tumors, you can see there is persistent vascularization. This persistent vascularization at the margins of the tumors is what causes tumor resistance, and this is called vascular normalization. Now, it's possible to look at perfusion of tissues using two different techniques: dynamic susceptibility enhanced MRI, which mostly gets used for the brain, and dynamic contrast enhanced MRI, which gets used for the body, and many of you will be aware of that. And so what we are looking at in a DCE experiment, the dynamic contrast enhanced experiment, is we are looking at perfusion and tumor permeability or vascular permeability. And there's a very interesting constant that quantifies that, and that's something called K-trans, the transfer constant. And the transfer constant essentially tells you about the blood plasma flow per mil per liter per second. Sorry. Okay. Now, so if you look at this particular patient who is getting an anti-angiogenic therapy, what you notice is that there is an anti-angiogenic effect. So this patient's getting Avastin, which is not licensed for this particular indication in GBM. You can see that post-contrast, we can see the enhancement go down. That decrease in enhancement represents the decrease in permeability. Now, if you look very carefully, there's an arrow here, which hopefully I can show you, there. Look at that area there. You see how that area disappears, whereas this area here remains. Can you see that? So you can see within the same tumor, there are areas where vascularization goes down, where the permeability goes down, in other areas where the vascularization doesn't change. So this is the phenomenon of vascular normalization that you can see in this particular patient. So we are beginning now to prize apart mechanisms of actions of particular treatments. Now, I was going to talk about metabolism, but I'm going to just skip that and just go to say a few words about applications. So we know, and we will hear shortly about multiparametric MRI being used in a number of different areas. So PIRADS, for example, for prostate cancer is one example. But you can also use it, and this is where I think it's most interesting, in looking at changes with novel anti-angiogenic therapies across time. So there are a number of challenges that we as a community need to face. Firstly, this is getting far too complicated. So many technologies, so many maps, repeated multiple times. This is driving us crazy. So that's where we are going to need artificial intelligence. So we've been talking about AI. Where is it going to be helpful? Right here, right? Multiple functional imaging modalities done multiple times. You know, if you do it 15 or 16 times, you lose track of what's happening. So I think that that's a challenge, and that'll be helpful. We need to develop a better understanding of what the hell is going on inside cancers using imaging. And this is the field of radionomics. Right? This is the field of radionomics. So you've heard about radionomics in this conference, but it seems abstract. This is what it's all about. It's about understanding what the hell is going on. The other thing is this heterogeneity. I already showed you a couple of patients where there was this heterogeneous response, different bits of the tumor doing different things. This is a nightmare. This is going to be a complete nightmare for us. How are we going to deal with heterogeneity between lesions, within lesions, and between different patients over time? That's 4-dimensional heterogeneity. Absolutely crazy. It's going to drive us mad. So here are my conclusions. Multiparametric imaging in the sort of space of precision medicine allows us to look at biological changes spatially over time. Okay, so it's multispectral, multi-dimensional. It can be used for a number of areas, and we will see that being demonstrated shortly, including response assessment. In precision oncology, it'll allow us to give the right treatment for the right patient at the right time and for the right duration, so we can switch treatments as fast as possible to allow exposure to as many medicines as we can for our patients. And we will need to develop other technologies that enables us to do this. Things like deep learning, artificial intelligence, which will help us not only in the clinic, but also to develop drugs. Thank you very much.